Hi guys, welcome to another instalment of Biology with Miss Cherry. Today we're going to have a look at the process of autosomal linkage. So could you please have a look at the diagram to begin with? What is happening in the picture on the left? So I'll put it in a red box for you. In the red box. And what is happening in the picture in the blue box? And why is there a difference in the products of crossing over? Now, if you're not sure about crossing over, you might want to look back to your notes that you did way back when in topic 4. Maybe you did it in the last lockdown. Hopefully you will have covered it. If not, there'll be a video somewhere on YouTube for you to recap and revisit that knowledge that you should hopefully have. Pause now, and then when you're ready, carry on and we'll get started with the lesson. So here we are, lesson 7.6. It's not a great, massive PowerPoint today. This is a very small topic that's quite detailed. So we're not going to spend too much time thinking about the fluffy kind of parts of linkage. Instead, we're going to take a look at how you guys will need to be able to recognise it, and that's in terms of observed and expected offspring. And this is going to link in nicely to when we look at the chi-squared test in a few lessons time. So as you can see on this presentation slide, we have got the title. We're going to describe autosomal linkage because that's a common exam question, and then we're going to see how that's going to affect our alleles in our gametes, but mostly need to recognise that it links to meiosis. So we're going back to module 4, as I said in the previous slide, and we're going to see how the spatial proximity of different alleles affects the genetic variation that we get in our offspring. So let's get started. So to begin with, we're going to think about what these keywords actually mean. So what do you think linkage means in terms of genetics? You might want to just think of the word linkage by itself. And then what do you think it's going to do to the phenotypic ratio of our offspring? So by phenotypic ratio, I'm talking about the proportion of offspring that look a certain way. So you can spend a couple of minutes thinking about that, or you can just carry on letting this video play and we'll go through that definition. So linkage itself is when genes are found on the same chromosome. Now, generally we say that they're quite spatially close, or they're spatially proximal to each other's. And if they're on the pairs 1 to 22, those autosomes that we've heard about already, we call them autosomally linked genes. Now, this is really important because today we're going to be looking at the autosomes. We're not going to focus on the allosomal linked genes, the ones related to the sex chromosomes. We're just focusing on autosomal linkage today. Now, if I've got genes on the same chromosome, they're likely to be inherited as one unit. If you imagine it a bit like if you've got a boyfriend or a girlfriend, you guys sort of linked together, you're going everywhere as a group. Okay, so if you've got invited to a party, if one of you got invited to a party, the other one's likely to come anyway, and there's unlikely to be any form of crossing over, any form of breaking during any stage of the party, maybe any stage during meiosis if we go back to thinking about the genes. Okay, so genes on the same chromosome are likely to be inherited as one unit if they are linked. Now, before we move on any further, we need to have a quick recap of Mendel's laws of genetics and all of the stuff that he discovered when he was doing all of his pea investigations. Okay, so let's take a look at those now. Now, through growing over 10,000 pea plants through his lifetime, around 8 years that he was actually studying this, he managed to find out these 3 laws of inheritance, and they were ignored for many, many years because everyone was like, no, religion is the way that things are the way they are. You know, that standard GCSE biology, why were people against things answer. And let's take a look at each of these in a bit more detail. So the first law is the law of segregation, or the law of random fertilization. Okay, and that is that inherited traits are defined by a gene pair. Okay, for us, we know that that's a pair of alleles found on the chromosomes. Okay, and these are, through development, gained from the random separation of genes into gametes, so in your mum and in your dad, and the subsequent random fertilisation of gametes. Okay, so you cannot, under any biological circumstances, decide which sperm goes with which egg. If you could, there's lots of ethical issues that arise with that situation. But random fertilisation is the random process of gamete fusion and subsequent fusion of genes. Okay, so we're talking about a random sperm and a random egg coming together, and the random genes, the random alleles that they've got in that sperm and that egg coming together to form you or another organism. Now, the second law is a really, really important one when we're thinking about this whole concept of gene linkage, is that the genes for trait A, where A is a certain trait, maybe height, are sorted separately from those for trait B, okay, where maybe B is something like eye colour. Okay, A and B have got to be characteristics, and therefore with this independent assortment, the inheritance of one trait is not dependent on the inheritance of another. So just because I've got a gene for small height doesn't necessarily mean I'm going to inherit the trait for green eyes, for example. The whole concept of independent assortment is that these genes A and B and maybe C and D further down the line are completely independent of each other. And the final law is the law of dominance. Okay, and this is a very standard, we know this already, or we should do anyway if these videos have helped you in any way, shape or form. The organisms with alternate forms of a gene, i.e. different alleles, will express the dominant form. Okay, and we know that if we've got a dominant allele, then that's going to manifest itself, and if we've got only recessive alleles, then that will manifest itself, but the presence of a dominant allele will always override the recessive. Okay, so to recap, segregation, independent assortment, and the law of dominance. Now keep those in your head for the next slide. And I want you to take a look at the image, okay? Assuming all the linked genes in the diagram are inherited together and passed into the gametes, think about those questions. So if we take a quick look at the diagram on the right hand side, we can see there's a section of the chromosome just here that are linked. Okay, and we've got gene 3 and the diagram highlighted to us that the genes or the area surrounding gene 3 are linked to it. So what that means is that gene 2 sticks with gene 3 and gene 4 also sticks with gene 3. And anything else that's in this space will stick with gene 3 as well. So with that in mind, think about which one of Mendel's laws, so we've got segregation, independent assortment and dominance, is being broken, and what do you think will happen to the observed phenotypic ratio compared to the expected ratio? And by expected ratio, remember we're talking about what we expect from a Punnett square. Because that's all the Punnett squares are giving us. They're giving us our expected ratio. And then play when you're ready. Now, as we said, if we've got our genes being linked together, they aren't being independently assorted anymore. Okay, so actually those linked genes are inherited as one unit. So remember in the example from the rules slide I said or gene A is inherited independently to gene B, maybe height and eye colour. In this case, gene 3 is inherited with gene 2 and gene 4, but separately to gene 1, because that's not in the gene linkage region. You'll notice that the things that are linked to gene 3 are spatially closer. And if you do OCR, or if you've got any friends that do OCR, they'll have to figure out the distance between genes. There's a calculation that you can do to figure out how far away each of those genes are, depending on lots of factors, things like the number of recombinants, which is what we're going to look at in a second. Now, compared to the expected phenotypic ratio, the way that the offspring look, there's going to be much more parental phenotypes observed. Now, on the next slide, we're going to look at this in terms of how to explain it using words, and then we'll do a diagram to show what we mean by the linked genes and the parental phenotypes and things like that. Okay, so bear with me. I promise hopefully it'll be understandable in a second. If you still don't understand, just ask a question. That's absolutely fine. Okay, ready? Here we go. During prophase 1 of meiosis 1, because remember, meiosis is made of 2 divisions, and prophase is where we get the condensing and sort of like the shortening and fattening of our chromosomes. Okay. Now, at this stage, non-sister chromatids of a homologous chromosome can cross over and break and reform. Okay, so you can see here that we've got 1 pair and we've got 2 pairs in the diagram, and at some point parts of the arms, so the chromosome or the chromatid arms, sort of twist around each other. So if they twist around each other, they're going to put strain on each other. Okay, so we're a bit like if we think about how temperature affects the structure of enzymes. If these little arms twist around each other, you can do it with your fingers now. Try and link your pointing fingers around each other. You get that kind of tension. Now below the break point, so after you've done the wrapping around and you've got that tension, any gene loci, okay, so the point of where the genes are found on a chromosome, any of those loci below the break will be exchanged. They'll be swapped over, and you can see that in the diagram there. So we've got a purple with a little b now and a little bit of the green chromosome, and likewise we've got a green chromosome with a part of the purple one. And these two now gamete chromosomes that have got different genetic combinations to the parents are called recombinants. Okay, so these are different genetic combination to the parents. Okay, now in terms of gene linkage, the closer gene loci are, okay, so we'll draw this on a diagram in a second, but if I've got A up here and then B and C, the closer they are, so B and C are quite close, the less likely that there is to be a break between those two genes. Okay, because they're so close, it's not really going to happen. Okay, so the closer they are, the less likely the break is between the genes, and therefore the crossing over of those genes is unlikely. And if you're not too sure about that, we're going to take a look on the next slide. So please still bear with me. If you understand, you're welcome to skip the diagram drawing section in a second. Okay. Now, if we start thinking about this idea conceptually, looking at the diagram, we can see that with non-linked genes, so there's quite a far spatial distance, or we say spatially distant, these two genes, A and B, are likely to be separated by crossing over. So you can see here there's the point of crossing over, and all of the genes below that point appear to have been crossed successfully. So if we get independent assortment, if we get that segregation, we've got a gamete produced by the left arm of the parental gamete. We've also got another unchanged gamete chromosome formed by the standard parental arm, as we can see there. And then we've got a couple of recombinants. Okay, so remember, these are the ones that are different genetically from the parent chromosomes. Okay, so you can see there that in non-linked genes, because those gene loci are so spatially distant, they're far apart, the arms of the chromosomes are able to cross over in the space between them. Okay, and that gives rise to those recombinants, that genetic difference. We can get crossing over of those genes because they're not very close together. However, if we take a look over at this linked genes example, you can see that genes A and genes B over here are very close together. Okay, now that means that where I've got this point of crossing over here, the genes themselves, A and B, are too close together for the crossing over between them to happen. So instead, although we've got some potential recombination, okay, we've got parental or different genes compared to the parents, they're not actually splitting the allele big A from big B, and likewise they're not actually splitting the allele little a from little b. So they're inherited as one unit. And we can say there that there's less chance of recombination. And that's what gene linkage is. Okay, so they're just passing on genes as one thing. It's almost like we can actually label these two gene loci A plus B rather than A and B, because they're always taken, or mostly always taken together. And as we can see here, we've just got another example, another bit of contextual information. Linked genes do not undergo that random shuffling, the random assortment. The linkage of the genes means that they're inherited as a single unit. Now we're going to take a look at an example of what you would expect from an exam question, and oftentimes that relates to body colour and wing length in flies. The Drosophila fruit fly is a really good example. It's quite small, easy to breed, genetically quite simple, and they're easy to keep and things like that. There's not a lot of ethical issues surrounding the use of flies. But we're going to focus on the two characteristics, body and wings. So we've got big B for brown body, little b for black body, big V for long wings, and little v for short wings. And as you can see here, I've not followed my own rules again, been a bit of an idiot. I've sort of shot myself in the foot because I'm using V's which look the same, both capital and lowercase, which is going to be a bit difficult, but you know, we'll deal with it. We'll have to, because sometimes in the exam they want to give you a rubbish letter to write. Now let's take a look at this example. We've got two parent flies, one of which with the genotype big B, big V, little v, and one with the homozygous recessive genotype 2 little b's, 2 little v's. Okay, now I want you to think, using the key, what are the phenotypes of the parents and what is the expected phenotypic ratio of the offspring? Okay, now that's a really wordy way of saying what do the parents look like, and you can use the key for that. And what is the expected phenotypic ratio? That's do a Punnett square. Okay, so pause and then come back when you're ready, and we'll show you how to do the answers. Okay, so we're using that key then to do the parental phenotypes first. And we can see that for parent 1, we have got genotype, and I'm just going to write it out in a nice format that makes it a little bit easier for me to do the next question. So big B, little b, big V, little v, and then we've got 2 little b's and 2 little v's. And we can see there that parent 1 has got a heterozygous phenotype for both traits, and for that reason we know that they are going to be brown body and long wings. Conversely, the second parent is homozygous recessive for both traits, and for that reason they are going to have a black body and short wings. Now, if I'm doing this in my standard Punnett square, sorry, that's question 1 done. If I do that in my standard Punnett square, I'm going to be finding out the gametes and then doing a cross. So I can see from that parental genotype of parent 1, I'm going to have to use my 1 3 1 4 2 3 2 4 rule. So 1 3 1 4 2 3 2 4. Put some commas in there so it actually makes sense. So 1 3 is big B, big V. 1 4, big B, little v. 2 3, little b, big V. And 2 4. It's like that. Whereas for our parent 2, our second parent, we've only got one set of gametes that are the same twice. So you can write it as you like, really. It's not going to make a difference. And then if I put that in a nice big juicy Punnett square, 2 3 4, and again for this one I'm going to be making sure that I'm writing in the phenotypes as I go. So we'll put parent 1 down the side, and parent 2 at the top, and I'm going to pause now. I'll pause the recording anyway and fill in the Punnett square because you don't need to see me writing letters, and then I'll come back when it's all done with the key. Okay, so we're back now, and you can see that we've got 16 offspring. I mean, you could have condensed this Punnett square and made it smaller, but we like to be thorough. And we can see here that we have got 4 brown long offspring in this row. So 4 brown long. We've got 4 brown short. We've got 4 black long. And we have got 4 black short. Okay, now this gives us a ratio of 1 to 1 to 1 to 1 on all of the phenotypes. Okay, so remember we're looking at the phenotypic ratios here. So this is the example of a smaller Punnett square that you could have done if you'd only done 2 of the parental gametes for the second parent. You know, they're the same, so why bother doing a big 4x4 one? I quite like doing a 4x4 one. It just works a little bit better in my head, but it does waste a little bit of time. Okay, so this is our expected phenotype, expected phenotypic ratio. Because we've done a Punnett square, and this is what the data suggests we should have. Now we expect to see that 1 to 1 to 1 to 1 ratio, but in actual fact, if I've done an investigation, I've done a study, and I've seen that there's a ratio of 5:1:5. So again, that follows the ratio of phenotypes shown in the brackets. So that's 5 brown long to 1 brown short to 1 black long to 5 black short. And I want you to thinking about what we said about recombination, thinking about what we said about gene linkage. Why are there more of the brown long-winged and black short-winged flies? You can look back to the parental phenotypes if you like. Hopefully it shouldn't be too tricky. Play when you're ready. And this is because of the whole idea of gene linkage. So if we take that parent 1, for example, and we draw out those chromosomes as if they were actually present. So we've got that, and then we've got another set of pairs. Right, so on the first set of chromosomes, we're going to have big B, big V, big B, big V. And on the second set of chromosomes, we're going to have the same genes, the same loci, but lowercase. Okay? Now, what we've got happening is that we're really mainly, even if we've got crossing over, going to produce alleles that have got the combination shown on the standard parental chromatids. Okay, so the big B's and the big V's stay together. And instead, we've only got a few of the recombinants formed by crossing over. So if you're not too sure what we mean by recombinants, go back to the slide where I drew the previous awful diagram. But by recombinants in this example, we only expect to see like a really few or really small number of recombinants that might look a little bit like this. Okay, because as we've said, these genes are really close spatially. The B's and the V's close spatially, which means that crossing over is going to be really unlikely. So again, if we take a look at the other alternative, and there we go, we can see again that little b, big V recombinants are also unlikely to be produced. Conversely, for parent 2, they are only going to be able to produce those little b, little v alleles. Right? And those crossing over, as we've said, of linked genes in parent 1, so the formation of these two is really rare, and those are our recombinants. Yeah, you can remember it saying they've recombined. Now again, we'll cover it again, we're going to keep doing it until it gets into your little brains, that the recombinants have got a completely different allele combination to either parent, which gives us that differing phenotype. So these would be our brown short-winged flies and our black long-winged flies. They're not going to look the same as the parents that they came from. In an exam though, you might feel a little bit pressured by time and things like that. So there are a few ways that you can identify and show linkage very quickly. Okay, so again, we're going to use the example that we had in the previous question. So we expect a 1:1:1:1 ratio of those phenotypes, but we observed a 5:1:1:5 ratio of the parental, and then a recombinant phenotype, and then another recombinant phenotype, and then the other parental. Okay, the way that the parents look versus the way that the blended phenotype looks. So if there are fewer recombinants compared to the parental phenotypes in the observed ratios of a genetic cross, it's likely that the genes are linked. Okay, and that's very visible in this example, because as you can see, we expect a 1:1:1:1 ratio, but actually in our parents, we saw that there were 5 of those offspring produced. So 10 out of 12 of the offspring were the parental type, or we expect 10 out of 12 offspring to be the parental phenotype. Okay, and there's much fewer recombinants compared to those parents as well. So only 2 out of 12 of those offspring are actually those recombinant phenotypes. Okay, so rule number 1, if there are many fewer recombinants compared to the parental phenotypes in the observed ratios of a genetic cross, it is likely that the genes are linked. Notice that we're using the word likely, not definitely. Okay, so if the genes are linked, the alleles for the letters stay together. Okay, so if we take that example, the genes for body colour and wing length in fruit flies are linked. What are the gametes produced by a heterozygous parent? And we use this example here. So if we've got the genotype big A, big B, little b, they are heterozygous, remember. Then big A and big B stay together, and little a and little b stay together. Okay, so if we know that the genes are linked, we only use 1, 3, 2, 4. We don't use the 1, 3, 1, 4, 2, 3, 2, 4, because in this question it has told us that they are linked, and that should raise flags. That should be a highlighted point for you straight away. Now for rule number 2, please in any exam question, assume that genes are not linked and will be independently assorted. So we do the 1, 3, 1, 4, 2, 3, 2, 4 until proven otherwise. So if the question tells you they're linked, or it gives you the cross to do at the beginning and then says, right, this is the actual observed offspring, are the genes linked? Then you need to assume that they aren't until you've been given reason to know otherwise. And it's not just a suggestion, you need to have that knowledge, that certifying the fact that they are linked, and then you can ignore the 1, 3, 1, 4, 2, 3, 2, 4 and just do the 1, 3, 2, 4. Okay, so now if you're in my class, could you please pause the video, have a look at OneNote and have a go at completing the tasks that I've put on there, the summary questions and how science works, and then let me know when you need the answers. So for the final bit of the lesson, as per usual, we're going to take a look at some exam questions. I'm just going to click through these and then we'll go through the answers at the end. Okay, so in fruit flies, the genes for body colour and wing length are linked. Explain what this means. So pause now and carry on when you're ready. And the next few questions are related to this stem. So a scientist investigated the link between the genes for body colour and wing length. Surprising, because we've spoken about this example already. He carried out crosses between fruit flies with grey bodies and long wings, and fruit flies with black bodies and short wings. And figure 1 shows his crosses and the results. G represents the dominant allele for grey body, and little g represents the recessive allele for the black body. Big N represents the dominant allele for long wings, and little n represents the recessive allele for short wings. And you can see in figure 1 up to where I'm putting the green line has all of the information that you need for that first cross. These offspring were crossed with flies homozygous for black body and short wings, and the scientist results were shown in figure 2. Okay, so we're using this information to do the next few questions. So I'll draw those lines on again. Use your knowledge of gene linkage to explain these results. So you're going to need to look at the offspring, you're going to need to look at the parental phenotypes, the recombinant phenotypes, and think about what you would expect using what you've learned today. So pause, play again when you're ready. It's 4 marks, it should take you about 6 or 7 minutes. If the genes were not linked, what ratio would the scientists expect to see in the offspring? And finally, which statistical test could scientists use to determine whether his observed results were statistically different from expected results? Now give reasons for your choice of statistical tests. If you're in my class, we have done this test multiple times. I need you to think back to when we looked at woodlice. I need you to think back to when you did your statistics lessons. And remember, it's a which test and give reasons for. Okay, and when you're ready, we'll start going through those answers. So for question 1, when it asked you about what does it mean for genes to be linked, you should have mentioned that those genes are found on the same chromosome. And that is to say that they're close together because they are found on that same short section of DNA that's wound up into that chromosomal structure that we know. For the next question, when we're starting to look at using our knowledge of gene linkage to explain the results, you should have suggested that, and this is where it comes into being able to use your words, that the big G, big N and little g, little n are linked. You think back to the question, we had a heterozygous parent, so big G, big N, little n, crossed with an offspring that was homozygous recessive for both. So G, or little g, little n, little n. Okay, and in that ratio, you should have seen that the offspring produced roughly a 5 to 1 to 1 to 5 ratio of the parental phenotypes, and 5 of the parental phenotypes for each type, and then fewer recombinants. And this is because the big G, big N, so remember the 1, 3, are linked, and the little g, little n, 2, 4, are also linked. And so then this parent here, this heterozygous parent, is going to mainly produce those big G and big N, big G, big N and little g, little n gametes. And again, we're going to get only a few of these recombinant gametes. And therefore, we're going to get fewer of those recombinant phenotypes. And it asked you about the genes, so you have to have used the alleles as well. Okay, and if you're not sure whether your answer is correct, if you're in my class, take a photo of it, type it out and send it to me, and I will give it a mark for you. The expected phenotype or phenotypic ratio is a 1 to 1 to 1 to 1 of all of the phenotypes. We've seen that before, we've looked at it today already in our previous example. And the final question is, what statistical test Should you use... It is the chi-squared test because it is categoric data. So we're looking at phenotypes. Grey long is a category. Grey short is a category. Black long, category. Black short, category. There's no range in those phenotypes. You can't be slightly grey with slightly long wings. It either is or it isn't one or the other. Okay, and remember, we're looking at the difference between observed and expected as well. And we did that when we thought about the movement of maggots during our taxis practical. Okay, so I hope you found that useful. If you've got any questions, let me know and I'll see you next time.